move on to slide 3. If a maple tree leaf cell has 14 chromosomes, what's the plant's diploid number? So again, we'll wait just a few seconds for everybody to think it through. And then I'll ask for responses. Alright, so what do you think? What's the diploid number?
I'm thinking of 7.
Okay, I can see where you're going with that. Let's define diploid. What does it mean to be diploid?
And I'm going to have to look that up. I mean, I know diploid and haploid are kind of the same ballpark.
Yeah, no worries. So, hey, welcome, Jesse. Our diploid is going to be our 2n, right? Our doubled. Haploid is the single number, or n. I like to remember it, haploid sounds like half the chromosomes. And di means 2. So with this one, if it's a leaf cell, it's not going to be a sex cell. So the diploid number is going to be 14. So the sex cells have half the chromosomes because they're haploid, but any other cell basically is going to have the diploid number. Does that make sense?
Yeah, I'm pretty sure I'll be
Okay.
able to.
Alright, so then if 14 is the diploid chromosome number, how many chromosomes would be in an egg for this same plant?
One egg for each chromosome? No.
It's just one egg, isn't it?
It is just one egg, so how many chromosomes are going to be in that egg? An egg is a sex cell, right? So what do we know about sex cells?
Gotta be 2.
No. 14.
I understand.
Okay, so let's let's see. Sex cells have just one N, right? They're haploid. So that means they have half as many as the normal cells.
Oh, 7.
Yep, so 7. That one was kind of a tricky one because you had to think that you had to think about the meiosis portion. And these were straight off of Professor Rosen's slides as some of her review questions, as are the next ones I'm doing. So these would totally be fair game in the test setting. Any other questions on these before we move on to the next ones?
Is this all on here so we can come back and review later?
It is on the slides and video recordings for review sessions. If you go onto the front page of the SI website, down towards the bottom you can click on that, and these will be up so you can review them.
Okay.
Alright, so here we go. In a human sperm, will there be any other chromosomes besides sex chromosomes? If so, how many? So this question's more talking about the makeup of the chromosomes. What kinds of chromosomes are you going to have in a sperm cell? Do you just have the X and the Y?
No, we have more than that.
They do. So if humans 2N is 46, right? Then how many chromosomes are going to be in a sperm cell? 23, yes. I like the little hands up symbol, Jessie. How do you do that?
I just clicked on it on the underneath our names on the left hand side. I'm not sure how to do this, so I'm doing my best here.
No worries. You're finding cool things that I don't even know exist.
Oh, I see it.
Oh, yep, there it is. There's your hand up.
Oh!
Hey, Jessie, we can see you now. Alright,
Yeah, I'm figuring it out.
I like it. So, in answer to your question on this side, humans have 22 single, like their haploid number
And
is, yeah, and one sex cell.
I got it backwards.
Oh, no worries.
I must be dyslexic tonight.
That's all good. We all have those nights. Alright, so... Oh, go ahead, David.
Oh, no, I was agreeing with you. We all have our off moments.
Yes. Alright, so what group of mammals lay eggs?
Oh, man.
This was on those questions too.
I think this was in the video somewhere.
Uh-huh.
Yeah, and it was in the PowerPoint presentation as well.
Yeah, all these questions from the beginning are going to be straight from the PowerPoint. So there's 3 types of mammals, right? We have the egg-laying mammals, the pouch mammals, and and the
Yes.
placental.
And these are the ones that lay eggs or... Oh, I wrote this down, I know I did.
No worries. If you need it from the textbook, I believe it's in chapter... Yes, it's in chapter 17, page 360.
Monotremes.
Monotremes, exactly. So the examples of monotremes that she gave were the platypi and
There's one other one.
I don't remember it though. Yes.
Bring back up the slides.
Oh, that was a terrible M, you guys. I'm sorry. I need a mouse. Yep. Monotremes. This test is probably going to be a lot on vocabulary, so be sure to know your vocabulary. Some good help to learn them and practice is your Mastering Biology. It's going to help a ton.
I totally bombed the last test. I've been having a hard time with this class.
It's a lot of material, and it goes fast, so I don't blame you. But we're gonna help you out.
It's really hard to remember. Even without any notes, it's really hard to retain all of them, especially when you start getting older, like some of us. It's even harder to retain.
Yeah.
I'm with you. Yeah, it's a lot, especially when you're, it's been 15 years since I graduated high school, so just kind of getting back into the study of school.
Yep.
15? I've been out 25, so yeah.
Welcome back. Way to go, you two.
Yeah, we're like totally non-traditional.
I had no interest in college when I graduated high school, but when I got my MD, my work every day, my research, yeah, hard to have for so long and use it.
Yeah, I actually know this one. Isn't it like marine animals? External fertilization? Like fish?
Yeah, so why would it be fish? What about their environment makes it nice?
Moisture, keeping it wet.
Exactly.
Dry out really wet.
Yeah, they dry out really easy, so they need to stay very wet. So we have fish that have external fertilization. Frogs was another example she gave. Pretty much anything that you'd find in a water type environment. Alright, so into genetics. If inheritance in rabbit color is due to incomplete dominance, what would the outcome be if you crossed a homozygous black rabbit with a homozygous white rabbit?
Spotted black and white, right? It's gonna dominate the little wolf storm, right?
That's codominance. That's alright. That's why we're doing this review, so we can figure out where we need to spend extra time.
Would wouldn't it be like a 50-50 chance of whether or not it would go black or white?
Actually, this is like the example she gave of the white and the red flowers, where the offspring were pink. Incomplete means neither one of them have control over the other. So you wouldn't have white, you wouldn't have black, but instead you'd have gray. An in-between color. Yep, so incomplete is in between. Then David with the codominance, you have both there, whether that's spotting or patches, whatever that may be. Alright, so if a couple has 5 daughters, what are their odds that the next child is going to be a girl?
I know this one. I'm raising my hand.
Alright, let's hear it.
50-50. There's no leaning one way or the other. You have 5 boys, you get the same chance to have a boy or a girl. Right?
Exactly. See, you know this stuff.
Yeah, so part of that chapter 7 video is in that question similar to that.
Yeah.
It's always 50-50 either way.
Exactly. It's always going to be 50-50 as far as what sex it's going to be.
Because the male has the X and Y chromosome, right?
Yep. Yeah, the female can only give X chromosomes because she only has an X and an X. But the male, he can either give an X or a Y, and it's 50-50 what it happens to be. Alright, this is on to chapter 8 material. I don't know if you've seen the week 8. I don't know if you've watched the videos yet, but I thought I'd include it just in case.
Oh, that's fine. I haven't got it yet, but it's in the cover anyway. Just watch the video later.
Alright, so if a strand of DNA had the base sequence CTAGGCTAT, what would the base sequence of the corresponding strand be? So,
Opposite of that?
yeah, the opposite. So, what we know about DNA is that the opposite of C is G. Always and forever. The opposite of T is A. Always. So in DNA, you're always going to see those pairs.
So it's the opposite of G, C?
Uh-huh.
Okay.
And the opposite of A is T. That is in chapter
10.
Chapter 10. Yep, you're right. Thank you. If you're looking for a page number that would have it, you go to page 174. Yeah, starts on 174. Matching is on 176.
Pretty confusing to try and read that. I'm still kind of lost. I was just reading that.
Okay, so with DNA, every segment of DNA has 3 things. You have to have a sugar, a phosphate, and then your base pair. And these letters right here are your base pairs. C's and G's will always come together, and then A's and T's will always come together in DNA. Now, the confusing part is probably when you throw in RNA. Am I correct? Yep. Okay.
Yes.
So, in RNA, which is basically RNA's whole purpose is to be a template. So, to create other things, other DNAs. And on RNA doesn't have a T molecule, right? It doesn't have thymine. It doesn't want it. Instead, in RNA, it's C's and G's together with U instead of T, matched up with A. And that's just because it has to have some way to be different from DNA, so you can tell the two apart. So, let's see. We're going to go into a little bit more detail on DNA replication towards the end. So, I'll leave you paused on that for that right here. So, in answer to the question, we have if it's DNA, then the opposite side would be GATCCGATA. And you just do whichever one's opposite. Does that make sense? Okay. So then we're going to take a step back and we're going to go in more detail with week 7 and week 8's lectures, and make sure we have all the vocabulary and everything down. So with genetics, there's a lot of vocabulary, a lot of vocabulary. So I thought it might be helpful to have a mixed and matched vocabulary list. That between the two of you, you can match up with its corresponding definition. So, David, let's have you do the first 4, and Jesse, you can work on the last 4. And then you can share with everybody.
Is there ever any more people in this session than this?
Usually I have a couple more. I don't know where Brandon or Shivani are. Usually they're both here. This might actually be the first week that Brandon hasn't shown, which is surprising.
I'm usually asleep already. I go to work pretty early.
Oh.
But I thought I'd try it since I'm having such an issue.
Well, we're glad to have you, and if you ever have any other questions, feel free to email me, because I know it's not a great time to do it, but
So do the last 4.
yep, you've got G genotype through heterozygous. How are your 4 coming, David?
I got 2 more left.
Alright.
Maybe like 2 more minutes.
No worries. Take as much time as you need. Can you two hear the background noise coming from my computer?
A little bit, not bad.
Okay.
That's kind of like static on TV a little bit.
Okay. If it gets bad, let me know and I
It's
can
not-
go
it's
out.
not that loud.
Alright.
It's not that bad.
Good. If it gets bad, let me know. I can go out and tell my roommates to quiet down a bit.
You should go tell my dad anyway.
I'll just go lay down.
Alright, I'm good.
Alright. What have you got, David?
Okay, dominant is allele expressed when paired with another allele.
Yep.
And polygenic trait is trait whose phenotype is influenced by several genes.
Yes. Where is it? Okay, there it is.
Yeah, number 5 down there. And then recessive is the allele not expressed when paired with another allele.
Uh-huh.
Second one from the bottom. And then Leo something or other.
Pleiotropy, yeah.
Pleiotropy, that one. It's on the first one, several traits controlled by one gene.
Exactly. So, the way I remember the difference between polygenic and pleiotropy, because they're pretty similar, is polygenic, if you break it apart, it means many genes. So you have a single trait that has many genes controlling it. Pleiotropy, plio kind of sounds like pliable, like flexible. So that one gene is really flexible in all the traits that it controls.
Thanks for remembering.
I know, right?
Yeah. Might seem weird to other people, but if it works for you, it works.
Alright, how's it coming, Jesse?
Well, the genotype is the physical appearance. Nope. The phenotype is the physical appearance, and the genotype is the genetic makeup.
Yep. So,
Then,
geno being the genes.
You know, being physical. You know,
I was trying to remember that one, because I was trying to remember the whole part of it.
Uh-huh. So we've got...
And I wanted to say that the homo was having similar, and the hetero was different. Is that correct?
Yeah. So homo, same. Hetero, different.
And I know this sounds crazy, but I was thinking homo is similar, same as one, and hetero as in 2 heterosexuals different. That's how I kind of remembered it.
Yeah.
I remember doing the homosexual thing.
Yeah.
I do the same thing.
That's how.
Those two are different. Yeah, I bet you everybody goes straight to that right away. That's probably the easiest. So
Yeah.
easiest to remember.
Oh my goodness, these drawings are not working out. Hold on. Oh no, I lost all the other ones. But I was gonna say, a picture of a homozygous would be like big A, little a, little a. And then a heterozygote would be big A, little a. Alright. So now we're going to practice some Punnett squares. Punnett squares can take a little while to get used to. They did for me. So let's go through this together. Oh wow, lovely square there. Alright, so the question, it's probably a bit small. Let me see if I can zoom in a bit. Is that better?
See back just a little bit. Yeah, just some of the work they're gonna cut off.
Is that better?
That's all.
Okay. So in humans, brown eyes are dominant over blue eyes. And in here it already gives you the lettering, right? That's what Professor Rosen said to do very first every time, is to give it a letter. So brown eyes are dominant, so they get the big B. Blue eyes get a little b. And we know that a brown-eyed man marries a blue-eyed woman. So what possible genotypes could the man have if he has brown eyes and brown eyes are dominant? Or
Big B, little. Yes.
yeah, it can be either one. I'll just leave it as a big B and a blank for right now. And he marries a blue-eyed woman. So what's the woman's genotype if she has blue eyes?
Little.
1 or 2?
2.
Do you agree, David?
Yes.
Alright. So we've got 2 little b's here. Let's see, they have 3 children, 2 of whom are brown-eyed and 1 of whom is blue-eyed. So we need to fill in this genotype and figure out what the man's genotype is going to be. So let's scoot it over a bit. So let's put the man on top. He's got a big B, and we don't know this one yet, so I'll leave it blank. And then the woman has 2 little B's. Which means there will be at least a big B coming straight down, right? And then we know that little b's will go across. Oh, was there a question?
How do you not know what goes up on the top right there?
Because there's 2 different genotypes that the man could be. He's either 2 big B's or a big B and a little b. So But what we're doing right now is trying to figure out if this is a little b or a big B. Does that make sense? Alright. So what are the phenotypes of these two children right here? Brown eyes, exactly. But if we look at the question, it says that they also have a blue-eyed child. So what do we need right here to have a blue-eyed child?
Small b.
Exactly, we need a small b up here. So it goes down. Here. So we have two children that are heterozygous, right? Heterozygous for brown eyes. And then we have two homozygous children for blue eyes. Sorry, brown eyes here, blue eyes here. I don't know if I said that correctly. So, let's look back at this question, see if we got it all. Draw the Punnett square that illustrates this marriage. What is the man's genotype?
B, little b.
Yep, big B, little b. And then the children's genotypes. What's the genotypes of their brown-eyed children?
Big B, little b.
Oh, what was that, David? You cut out.
I agree.
Okay, yeah. So big B and little b. That's what they have to have. What I'm going to do... What was that?
Oh, I bet you, I was guaranteed that one of these is going to be on the exam.
I'm guessing you one of these will be on the
She
exam.
Yeah, she'll have one on the exam.
Yeah. So, how about this? Where did it go? I'm going to give you guys another example, and I'm going to have you two do it on your own, just on any paper you have, and then let me know. So, let's see. Come on. Where in the textbook are the Punnett squares? Do you guys know? I'm trying to find them.
Round 152, I believe. Well, that's the Labradors anyway.
Oh, yes, yes. Chapter 9. So,
148.
148, thank you. So I'm going to give you an example. And you let me know. So let's see. If... You know what? I had one on my other page. Hold on one second. So you perform...
Flip the screen over a little bit.
I can see that one, yeah.
Yeah, see the wording.
Yeah.
I'm trying to set up my square.
No worries. Does that help?
That's fine. Thank you.
Okay.
gonna put it on the top like the man.
Mm-hmm.
And then green is the recessive, so it's gonna go on the side.
Mm-hmm. Yeah.
And the big yellow is gonna come down on both sides.
Yeah, so we know that the green has to be 2 recessive ones, right? 2 lowercase letters.
Yep.
What we need to figure out is, like this one that we used with the brown eyes, is the yellow parent homozygous or heterozygous? Does it have 2 big letters, or does it have a big and a little?
It has to have 2 big, right? 2. To get yellow all the way across, it would have to have 2 big Y's at the top.
Exactly. Mm-hmm. Yes.
Yep.
Yep, so your Punnett square should look something like this.
You're gonna have 2 big Y's at the top, 2 little G's side, and you're gonna have 4 squares that are big Y's with small G's, right?
Yeah, that's what I got.
Yep. Yeah. Alright, so I'm going to take this one step further. I'm glad you have your confidence back. You got this. So I'm going to take this one step further. So now that you have all the children being a big Y and a little y, or a little g if that's the letter you used, what if you cross 2 of the children? What are the genotypes of their children going to be?
What would be?
So
Does
again,
that work? Can you do YG?
you can do YG if it's a simple 1 trait Punnett square, but if it has more traits, it might get confusing to use different letters. So I just use the same letter, uppercase and lowercase. So you want to set up your Punnett square just like before, only this time you're given what goes on either side. We have a big Y and a little y or g, doesn't matter in this case, on both sides.
So the top square is 2 big Y's, and then a big Y, little y, big Y, little y, and then 2 little Y's. Right?
Is that what you got, David?
Yeah, it is.
Alright, yeah. So we're going to do just the same thing. You take the big Y all the way across. And with this one, you're going to take the little y all the way across. Oh, I did that backwards, sorry. But a big Y and a little y. And then this one has a little y